Aromatic L-amino acid decarboxylase, called AADC, is an essential enzyme required for synthesizing dopamine, which moderated cortical input in the putamen. In patients with AADC deficiency, mutation in the dopa decarboxylase gene causes profound loss of dopamine in the putamen. The primary phenotype, including a developmental delay, Dystonia, and movement disorder, particularly limb and truncal dystonia, and ocular crisis. Recently, an associated viral vector mediated delivery of the dopa decarboxylase gene into the putamen was shown to providing a transformative medical benefit across various patient ages, genotype, and disease severities. We have earlier demonstrated marked improvement of both cognitive and motor function, disappearance of painful dystonia, and decreased occurrence of ocular crisis. However, to date, there is no clear information regarding a neurological mechanism underlying the beneficial effect on the motor system after local replacement of dopamine in the putamen. In the motor system, cortical signals of motor intention for the prefrontal cortex are transmitted through the premotor cortex and the supplementary motor area to the primary motor cortex for the complex motor execution. The subcortical region receives this cortical input and sends feedback out to the cortex, which forms the cortico-basal ganglia network. The putamen targeted by AADC gene therapy is a hub for the cortico-basal ganglia network, which comprises a functional circuit that have been implicated in the various cognitive and motor processes. The putamen area can be topographically divided into the organized subsection connected to the cortical areas. Therefore, it has been implicated that the therapeutic effects on the motor system are associated with a specific transduced putamen area that belong to the distinct cortico-putaminal networks. We examined neuroimaging data of 8 patients with AADC deficiency who received AADC gene therapy. To evaluate the transduced putaminal area, we used a high spatial resolution PET with a specific AADC tracer, FMT. Group analysis of FMT-PET images showed that uptake of the FMT in the putamen was significantly increased from baseline to the 6 months post-treatment and has been sustained for 2 years. The spatial distribution of the FMT uptake in the putamen and 3D model showed that the highly transduced putamen area has been sustained at the center of the vector injection location for 2 years. The persistent expression of the AADC in the specific putamen area over the years indicate a constant dopaminergic restoration in the distinct cortico-putaminal network. In addition to the FMT-PET images, using the structural connectivity-based persuasion and 1,000 cortical regions of interest belonging to the 7 cortical networks that were associated with the striatal function properties, we calculated the volume of highly transduced cortico-putaminal connection areas. First, we investigated the possible association between the motor performance and the highly transduced putaminal areas connecting to the cortical areas in the motor system. We found the prefrontal cortex and the premotor cortex were primarily connected to the highly transduced putamen area across patients for 2 years, and the highly transduced prefrontal cortico-putaminal connection area was significantly associated with motor performance at 6 months post-treatment. Next, we examined the cortico-putaminal connection areas of the 7 striatum-related cortical networks. We found that the highly transduced prefrontal cortico-putaminal connection area of the frontoparietal control networks also exhibit an association with motor performance at 6 months post-treatment. From the perspective of the putaminal AADC gene therapy for Parkinson's disease, the central mechanism underlying the motor function recovery by the putaminal AADC gene therapy has been assumed to be a restoration of the motor cortico-putaminal network to recover motor executive function. However, our results demonstrate that the persistent dopaminergic restoration in the prefrontal cortico-putaminal network is a basis for the therapeutic effect of the AADC gene therapy on the motor system. It provides a novel insight into the mechanism underlying the motor improvement by the putaminal AADC gene therapy. Since the prefrontal area contributes to the cognitive motor control function, including motor initiation and planning, our results suggest that putaminal dopamine promotes the development of an immature motor control system, particularly in the prefrontal cortex.